Okay, so we'll start now. Just a little bit. Okay. Sorry, just need to arrange the window a little bit. Okay, today we are going to talk about the synapse. So the principle of the synaptic transmissions and what the the structure and how we talk about the neurotransmitters and so on. Let's start. So as I mentioned in the first class, there was well, debate on how the neuron actually connect each other. Okay, and at the very beginning, the time, the most simple idea is that how the neurons are being connected, and there were two possibilities. One is the neurons are directly connected physically. Okay, it's just like wire. Okay, and another idea is actually that they are not directly connected to each other. So there's a small gap in between. But because at the very beginning, the limitation of visualization of those structure, it was not quite sure. Okay. However, some of the development in which that in the earliest, some of the experiments, classical experiments by scientists as early as by Dr. Bennett, and as early as 1851, the simple experiment using the frog nerve and muscle. Now, at the very beginning, they are mostly detected using, the animals. A frog is one of the one as an experimental animal. The reason behind is actually even you kill the frog, then the muscle can still move at some time. And if you go to the live market and you see how those people, sacrifice those frog for their meat, and then you can see that actually after chopping off the head, the muscle can still, move. In which that in this experiment, the scientists actually get the muscle and then still have the nerve muscle preparations, and then examine a chemical called curare. Curare is actually used for hunting in South America. Is a toxin from the plants. So you see that a lot of toxin or poison from plants or animals are actually making use of those. So for example, if the plants can secrete this kind of toxin, then the animal will not eat it as a food because it's toxic. But but people use that for the hunting because that you can put it in the arrow and then shoot it to the animals, and then the animals will not be able to move. Now, actually, this is a chemical that blocks the transmission. Blocks the nicotinic acetylcholine receptors, in which that receptors actually contribute to the transmission of the signal from the nerve terminals to the muscle. Now, when Dr. Bennett actually showed that when the muscle was bathed in the solution containing the curare, it was not able to stimulate the nerve muscle, and the muscle contract. So that means that there's something that blocks the transmission. So if it is physically connected to each other, then that is not the case. Even if chemicals will not be able to, affect the transmission. So this is some of the not very direct evidence that there's a synaptic transmission presence. Okay. Now another experiment, very famous actually, is about the heart. Okay. It is first described by Otto Louis in 1921. This is a more conclusive evidence. Now, the heart actually is innervated by both the sympathetic and the parasympathetic nervous system. Okay, in the parasympathetic system, if you still remember, that's innervated by the vagus nerve. Okay, so here you can see that. Now, what happens? The activation of the parasympathetic system will slow down the heart, the beating rate of the heart, okay, which is different from sympathetic system. Okay, so this experiment is actually very simple. It happened again in the frog heart, as again the frog tissue are more resistant to stop, so it's as I mentioned, the muscle or even the the frog still can survive for some time. So what happens is that in this very common kind of setup, there's two chambers, and then they connect to each other by a flow of buffer, cyanide buffer, to keep the heart alive. You can see that there's some air bubbles, so that the heart can still survive for some time. So what happens is that the scientist actually stimulate the vagus nerve. Okay, and what happens when you stimulate the vagus nerve, that actually slow down the beating of the first heart. Okay, there's two heart. There's a so-called donor heart. There's a recipient heart. Okay, so when stimulate the vagus nerve, it causes the donor heart to slow down. Okay. Now this is something actually may be lucky or something that, because in the parasympathetic system and the sympathetic okay. Now, but another interesting stuff that he observed is that the second heart, after some time, they also slow down. Okay. So that means that according to this, the Louis postulate there is some substance actually released from the nerve ending, and then travel through the chamber, the connections to the next heart, and then cause the effect. Okay, and that is actually the very beginning that actually they find out, that actually chemical is actually the chemical is actually acetylcholine. So this gives the very first evidence that actually chemicals are released from the nerve terminal and then transported to the tissue, and then initiate those actions. Okay, so there's evidence that there's a synaptic transmission in the nervous system. Okay. So later study actually found out that that is the synapse. Okay, synapse synapses are actually specialized site where the neurons communicate with other cells. Sorry. Okay. Now, so there's an axon terminal from the presynaptic cell. Will send the signals that are picked up by the postsynaptic cell. Okay, presynaptic cell and then postsynaptic cell. We'll look at those in more detail. There basically have two types of the synapses. The first one is the electric synapse. The second one is the chemical synapse. Okay. So they are actually quite different, as you can see. Imaged in the electrical synapse, there's a gap junction in between, so allow the current to pass through. Okay, they are very close together, almost touching, but there's a small gap, so-called the gap junctions, allow the ions to pass through. Okay. Now, whereas in the chemical synapse is a bit different, there's no intercellular connections. So no channels that connecting together. There are proteins that actually hold them together, but there are no direct connections. Okay. In terms of the intercellular connections, but there are outside. There are proteins that link them together. Okay, we'll talk more about that later. Now, what happens is actually chemicals are being released, and then the so-called the neurotransmitter, and then that will diffuse to the postsynaptic cell, and then activate the receptor. Okay, so there's a chemical differences. Now, in those hard experiment, or in the first experiment about the curare, you can see that actually in the curare that prevent the neurotransmitter binding to the receptors, whereas in the heart experiment is the release of those neurotransmitters that diffuse it all the way to the receiving heart. Okay, so those two are the example how the experiment provide evidence that actually this is what happens. Okay. So we first talk about the electrical synapse. So here is the what the structure is. This is the presynaptic cell membrane. This is the postsynaptic cell membrane. Now they are connected through the pore, so-called the connexons. Okay. Those connexons can be closed or opened. So they are hexameric unit. Okay. As both in the pre and postsynaptic terminal. Now, if you look at the transmissions, this is a presynaptic neuron when the action potential arrive, and then there will be a short delay, and then there will be, another action, the current in the postsynaptic terminal. Okay. Now, this kind of transmission is very fast because there's no, almost no delay. There's really brief delay, almost no delay at all. So that action potential can transmit in very fast fractions. For example, in the crayfish. Okay. Now, the advantage of the electrical synapse, as you can see that the gap junction is very, very narrow. Okay, around 3.5 nanometer. That has to be the case because otherwise that you know, that will not be enough to transmit the signal, okay, to the other end, okay. Now, another very important idea is that the synapse is not necessarily transmitting in an action potential, particularly in the chemical synapse, okay. Those are changes in the potential, membrane potential in the neuron. So this is a very important that you have to remember, okay. In some of the cases that will be action potential, for example, in the crayfish nerve. So you know what is crayfish are? The small, like freshwater lobster. So when they escape, you know, they flip the tail, that is through the electrical synapse, okay. So because when they escape, they need to be very fast. Otherwise, they will be caught and eaten, okay. So that's in the really primitive animals, okay. Now in the more advanced organism, for example, in vertebrate, in some of the cases, that is being used to synchronize a group of neurons, so that they will do the same thing together. So one of the examples is the hormonal secreting neurons in the within the hypothalamus. Okay, they are connected by electrical synapse. Now, this arrangement allows them to synchronize together because, you know, if you have studied the in the physiology, the hypothalamus together with pituitary actually the one that releasing a lot of hormones, and actually a lot of those hormones are produced in the neurons. And actually, when the time that we talk about neuropeptides, then you know that a lot of those neuropeptides can serve as the hormone in the peripheral, and but then they will so one of your classmates asked, are those channels highly selective for ions? Not quite, actually. When we talk about it, actually other molecules can also pass through. Okay, not just the potassium or sodium. Okay, they are not very selective. Yeah, so they are not like some of the selective channels. They are more like, you know, a passage of channels. You know, not selective compared to, for example, the potassium ion channels. Okay, this is a little bit different. Very good. So you get the idea. Okay. So as mentioned here, the synchronizing the cell for facilitate the hormone release, the secretions. The pores are large enough to allow molecules such as ATP or secondary messengers to diffuse it, but not necessarily big proteins though, okay? So, do electrical synapses ensure a single directions of the signal transmission? Not now for the ions. They actually can sometimes they can go to both actually because it's a open, you know, open channels. But because of if that is the action potential passing through, then that is still one direction because the other side will still have the you know, unilateral directions of the flow because of the refractory period. Okay, but certainly the other chemicals can pass through in both ways. So the cell actually kind of, you know, connect to each other. Okay, so then actually it's one of the not very good features as well. So that because those cells will be, you know, can affect each other directly. So does the direction lead by depolarization? No, because of the refractory period, you know, still applying to those. So the chemical synapse. So chemical synapse is more specific, more special visualized it, so-called and for example, one of the type, the most common type is exodendritic synapse. That is actually formed by the between the axon terminal, which is a presynaptic terminal, and the dendrite, which is a postsynaptic terminal. So if you still remember the last class, we talked about the neurite. So there are axon, terminal, and dendrite. Okay, so those are the one image that in the exodendritic synapse, the terminal actually, axon terminal is actually synapsed with the dendrite. Okay. Now the cliff is actually a little bit, the synaptic cliff in chemical synapse is around 30 nanometer. So which is quite thick compared to the electrical synapse. Okay. So there's also have time, well, quite time you needed for the molecules to diffuse that cause the synapse. Now the action terminal actually contains mitochondria, spherical synaptic vesicles, and then projections on the postsynaptic membrane. So we'll look at the picture and we'll see that actually those proteins are linked together, so that they are not just floating around, they are actually linked together. So there are proteins that link the pre and postsynaptic membrane. So actually when we talk more in detail, you'll see that actually the chemical synapse is not just that simple. In order to have one, both partners needs to be committed. Okay, it's not just like the postsynaptic terminal is controlled by the presynaptic terminal. It's not actually both. Actually, in order to function, both terminals needs to have a closer relationship. Okay, so it's bi-directional communications. Okay. So here is some of the more simple illustrations of that. You know how the chemical synapse. Okay, so basically what happens? Axon with the action potential arrived, and then that will stimulate the release of the neurotransmitters over here, and then the neurotransmitter will bind to the receptors, and then that you'll have the signal transmitted from presynaptic terminal to the postsynaptic terminal. Now in the synaptic cliff, actually is that's 30 nanometer gap. There are proteins actually linking the presynaptic terminal and the postsynaptic terminal together. Okay. Now here is another example of it. Now you can see that there's this is the neuron, this is the axon, one of the axon, and this is the axon terminal, presynaptic terminal. This is the postsynaptic terminal. Okay, this is the so-called the active zone, in which there are a lot of small synaptic vesicles. Okay. Now there are other vesicles as well, which is called secretory granules. Okay, you can see that the secretory granules is around 100 nanometers. They contain maybe peptides and proteins. Okay, the synaptic vesicles actually is smaller. It is around 50 nanometers. Okay, and those contain the neurotransmitters. Okay. Now you may ask why there's mitochondria, because that actually this process actually require a lot of energy. When we talk more in details and how actually the vesicle are being released, then you see that actually that require a lot of energy. Okay. So one of your class may ask, what is the function of the secretory granules? There are different functions, but I will talk about that in more detail in later slides. Yes, just remember there are two types: synaptic vesicles and then synaptic granules. Now, classification of this chemical synapse. So you can see that actually we always classify those for the ease of comparison and remember as well. Now they are classified according to where they are located on the receiving neurons. For example, the azodendritic synapses make that the synapses are made from axon to the dendrite. The other one is axosomatic one. Is the axon terminal on the cell body. The axonal one on the axon, so axon to axon. Okay. The dendritic on dendrites to dendrites, but this one is actually very rare. Okay. Major of those, a lot of those are actually exodendritic. Okay. So here is the example. So exodendritic, so axon on the dendrites. Okay. This one is axosomatic, that you in the cell body. This one is axonal, so you can axon to the axon. This one dendritic, which is quite rare. Okay, now you can see that the synapse on the cell body are often inhibition. Synapse on the dendritic spines are more often excitatory. Okay, now axon to the axon, that's usually modulatory. Okay, so eventually you can modulate the release of this axon. Okay. Now, some of the comparison between these two types of synapses, electrical and chemical, certainty is greater. In the chemical is smaller. So why certainty? Because that the electrical is always going to pass through the signal, but the chemical will depend on the chemical transmissions and the modulation as well. The lag period, is really fast. For the chemical, there is a lag because of the diffusion of the neurotransmitter across the synaptic cliff, so that is much slower compared to the electrical synapse. Now, chemical messengers, as we mentioned, the ions and small molecules can pass through the gap junctions so that the neurons can communicate it. In the chemical synapse, the most important is the neurotransmitters. Okay. Magnifications. Magnification is much bigger. So what does this certainty means here? Certainty means that actually, so when you transmit the signal, what is the probability of successful transmissions? So in the electrical, actually is much quicker. So because it's not involving any chemicals, diffusion, and so on. You know, but the chemical synapse actually depends on the integration. For example, there's when we talk about the synapse, actually there's an inhibitory and excitatory, and that can cancel each other. Okay, so when it's being cancelled, then there's no initiation of the action. So that may not be a bad thing, actually. Okay. Magnification, the electrical synapse actually is much smaller, so it's more like a direct. Now, compared to the chemical synapse, actually that's much greater, because you can, if that can, because the chemicals actually can activate is more advanced form, so it's more common in the vertebrate. For example, most of our synapses actually are chemical synapses. Okay. So when it so one of your classmates asked whether certainty is the effectiveness of the presynaptic membrane to activate the postsynaptic membrane. Not quite effectiveness. So let's say that the certainty means that when you transmit a signal from one neuron to another one and result into the activation of the next one, that's higher. Not quite effective, there's higher probability to activate the postsynaptic neuron. Okay. Now, but for chemical synapse, actually, that depends on the integration of the signal. Is it okay? I will not say it is effective. Effective is something that you will say that the electrical is better than the chemical. That is not the case. It's a certainty. It's okay. Now, neurotransmitter, neuromodulator, and neurotrophic factor. This one is also a bit maybe. Let's talk about it first, and then if you are not sure what does it mean, then we can discuss it a little bit more. Because usually students will be a bit confused by that. Neurotransmitters are the one that release about the presynaptic terminals and produce a rapid excitatory or inhibitory response in the postsynaptic neurons. So you can see that it's excitatory and inhibitory, both are possible. Now there are known neurotransmitters that we are known. Another putative neurotransmitter. Biogenic amines, catecholamines involve the epinephrine, norepinephrine, and dopamine. Now you may ask that the epinephrine or norepinephrine, actually those are hormones. Now remember, those are secreted by the adrenal gland. Actually, adrenal gland is also containing a neuron-like cell. Actually, those are in the sympathetic nervous system. Those are also neurons secreted. So you can see that a hormone and neurotransmitter, actually they are quite similar in the sense, but those in the brain will be used as a neurotransmitters, but in the peripheral that seems to be the more the case of like in hormone. Now endoamines include the serotonin and histamine. Some of those we'll talk about those in more detail in the later section of the course. Ester is acetylcholine. That one is like the peripheral muscle, and also in the central nervous system, that also used as a neurotransmitter. Amino acid, for example, excitatory, glutamate is excitatory amino acid in the neurons. Inhibitory, the most important one, the gamma aminobutyric acid, short form GABA. We'll also talk about that in more detail. Actually, GABA and glutamate, actually, those are the two most important neurotransmitter that our cognitive functions. Okay. Now peptides, substance P, and caffeine, somatostatin, and neuropeptide Y. Peptides are actually short linkage of amino acid, maybe 7 or 8 amino acids links together to become peptides, and they can be neurotransmitters. The other one, including gas, actually, you might you might not imagine gas actually can form as a putative actually neurotransmitter. Nitric oxide is the one that actually we know, we study it quite a bit. We will talk about that in more detail in the course. Carbon monoxide, actually, some people postulate that actually that can also form as a neurotransmitter as well. Okay. Okay, so neuromodulators, they are the released by neurons and astrocytes and produce a slower pre or post-synaptic responses. Usually they're mediated by G protein coupled receptors. So they modulate the response of the neurons. Okay, they are released by neurons or astrocytes as well. Other neuromodulators, including the nitric oxide, yes? So one of your classmates asked that the chemical can be both a neurotransmitter and a neuromodulator. Yes, actually depending on what's the action on it or how you define it, you know. So neuromodulator means that it modulates the activity of the neurons. Okay, it depends on how it works. For example, if it initiate a membrane potential changes, then that might be a neurotransmitter more than a neuromodulator. So there's a bit of, you know, how do you say, is a bit of both, you know, depend on how you define neuromodulations, but that is also right, depend on where it is, yes. So they may be produced by non-neuronal cells as well as neurons. So you can see that at once that you see even in the non-neuronal cell, they can secrete neuromodulators. For example, in the astrocyte, they can produce those. Okay. Neurotrophic factors. They are, for example, tyrosine kinase linked receptors. They are actually regulate gene expressions and encourage neuronal survival. Now, neurotrophic factors is very important in the survival of neurons, particularly in neuron and muscle, neuron and neurons. Okay, because that, you know, this is something, is like, you know, a relationship in which that if you want to support a relationship, then you have to give something out to encourage, you know, just like you and your boyfriend, or you and your, or not just boyfriend or girlfriend, it's just friendship, you know. When you like hanging out with somebody because they say some reward, happiness, you know, that both contribute. So in this case, the neurotrophic factors actually is like that, to encourage the survival of that. If without those encouraged, then the connection will disappear, and then the relationship will end. Okay, that is actually very important. So when we talk about the receptors, then you will see that actually those are the trophic factors, so-called. Okay. Now, apart from those, there are other mediators as well. For example, cytokines, chemokines, and growth factors. They all have a long-term effects on the changes in the brain, steroid as well. You know, for example, the synaptic plasticity and also the remodeling, those are required. Now, those are actually a lot of time they will cause affecting the gene transcriptions. So whenever there's a gene transcription changes, there will be a long-term effects on it. Okay. Is it okay so far? So it's more like a definition of it. Now, so as we mentioned, the nervous system is developed as a Neurotrophic factors affecting our memory. Oh yes, the neurotrophic factors actually will affect our memory, and actually that also can affect a lot of things. For example, our happiness. And when we talk about, for example, in the case of depressions, that actually are part of the reason is actually the lack of the neurotrophic factors. Okay. Yes, very good actually. All of you can think of those effects. So that's the reason why that when you are happy, you get more neurotrophic factors, and then it will benefit your brain. Okay. So back to this neurotransmitter. So what for as neurotransmitters, and actually you can see that actually the evolution actually selects some of those. As we mentioned, the evolution of the nervous system is because that actually the organism needs to detect the environment changes, and so that's actually how that evolved. And certainly those neurotransmitters are some of the medical important basic chemicals important in our system. So so that's also the reason why that from cells and species to bacteria, even giraffes, and those chemicals in the metabolism are actually used by all the organisms. So interestingly, one of your students asked, isn't happiness caused by dopamine and serotonin, which are neurotransmitter? Partially, yes. When we talk about dopamine and serotonin, then we will see. But happiness or your mood is not that simple. Just depend on one chemicals. Is again, it's more complicated, and certainly we don't 100% understand the mechanism, but we know that actually the neurotrophic factors, the neurotransmitters are related. Yes, that's for sure. Okay. So back to this. So particularly, for example, amino acid is the building blocks of protein. So that being used as a neurotransmitters, but that is expected. For example, is why the reason that actually we want to detect, the building blocks of the proteins. Okay. So most of the known neurotransmitters are as follow: amino acid. Amine is derived from amino acid. Okay, and peptides as well, which is derived constructed from the amino acid. So you can see that the amino acid actually is very important. One exception is the acetylcholine, which is derived from acetylcoenzyme A. Okay, it's a little bit special. This one is actually more related to the metabolism. Okay, but a lot of those are actually, when we talk about the other amino acid or a neurotransmitter, a lot are from the amino acid. Others are derived from amino acid. Okay, for example, dopamine and serotonin, actually they are neurotransmitters, and also they are derived from amino acid. And when we talk about those neurotransmitters, then we'll discuss that in more detail. So Dell's principle. So this one is actually one of your classmates, I still remember, asked about whether the neurons actually use one neurotransmitter. Now, initially, they so-called a Dell principle. So a neuron has only one neurotransmitter. So Dell principle is actually proposed by Dell, which is a neuroscientist. The one which state that the neurotransmitter are generally stored and released in the one type of neuron. Okay, So that's the reason why that some of those called cholinergic, glutaminergic, and GABAergic. However, later it was found that that's not quite true. So the neurons actually use more than one neurotransmitter. Now, in which the other neurotransmitter is actually the peptide containing neurotransmitters. Okay. So, for example, if you still remember earlier in the class, at the beginning of the class, we talked about the vesicle. There are small synaptic vesicles that store the neurotransmitter. There are large, dense core granules that store the neuropeptide actually. Okay. Now, during the transmission, if there's low frequency, that only preferably release the neurotransmitter from the synaptic vesicle. Okay. Now, however, when there's like really high frequency, remember the neurons encode the information through the action potential in the case as frequency. So low frequency and high frequency means the high signal, and in that case, then that will be enough. Then both the neuropeptides and the neurotransmitter will be released. Okay, so by the time that we talk about the synaptic granules, those are the ones that actually contain the neuropeptides as well. Is it okay? So some more classifications. There are fast form of the synaptic transmission that include the glutamate and GABA as well. Fast means that the transmission is actually the action will be fast. When we talk about those, then you see that actually those are because of the ion channels are involved. Okay. Now there's no form of the synaptic transmissions. Actually, most neurotransmitters actually are include the slow form of the synaptic transmission. Those are more involved in the G protein coupled receptors. Okay. Now, some of the example here is the mixture form of the synaptic transmissions, both fast and slow. That actually involve the acyclovir, for example. Okay. Some of those are coexist in the same neuron. For example, peptide plus amino acids or peptides plus amines. Now they are released under different conditions. So, for example, here is the case. There's low stimulations. That's the neurotransmitters. In the high frequency, even the neuropeptides are being released. Okay. So, in the case of the definitions of the neurotransmitter for the study of the neurotransmitter, usually people like to define what do we mean by neurotransmitters. Okay, so just like what is a neuromodulator? So to as So the peptide facilitate, amplify the release and binding of the binding of neurotransmitter. Not necessary. It may have other functions actually. When we talk about the neuropeptide section, then you know actually they are not necessarily facilitate or amplify. They may have other functions. Okay. Back to the criteria for the neurotransmitters. So there are at least three criteria. The anatomical, so that must be there. So this one is very, very straightforward. So it must be in the target area being released. Biochemical, so they must be synthesized, released, and removed. So when we talk about individual neurotransmitter, you'll see that actually both those are very important aspects biochemically, okay, of the neurotransmitters. Physiological and pharmacologically. So the identity of the action. So the neurotransmitter, when released, it must be have the outcome. Okay, for example, the release of acetylcholine at the neuromuscular junction will lead to the contraction of the muscle. Okay, that's the actions. Okay, and some chemicals can also mimic the effect. This is a so-called pharmacological. And any chemicals that modulate the neurotransmitter. What does anatomical means here? One of the classmates asked. Anatomical means the location. Okay, so for example, if you say that the acetylcholine is a neurotransmitter in the causing the muscle to contract, so those are terminal of the nerve terminals at the muscle must contain the acetylcholine. Is it okay? So that's anatomical. Okay, so just the location, which makes sense. You know, if the action is there, then they must be there. If they are not there, how can they be the neurotransmitter? Okay. So here's what means by anatomical. So the substance must be present in the nervous system in a reasonably concentrations. For example, if you look at the brain, they will contain all those neurotransmitter in high concentrations. So it should be present in the synaptic vesicles of the nerve terminals. So at the nerve terminals, there are synaptic vesicle containing those neurotransmitter ready to release them. It should be localized to specific pathway in the brain. When we talk about individual neurotransmitter system, then you'll see that actually they're forming by so-called nuclei and then going to different areas. You know, those are the ones that we talk about. Different nuclei, actually, they might contain different neurons that secrete different neurotransmitters. There should be a drop in concentrations of the substance foreign lesions in the substrate pathway. So this is also simple. That means that actually lesions mean the damage of those neurons or that group of neurons. Then because of the damage of that group of neurons, then those neurotransmitters, those concentrations will drop. Is it okay? So that means that those chemicals are synthesized by that group of neurons. Okay. So that's anatomical. Biochemical. So there are enzymes that for the synthesis and the degradation. So there are metabolic pathways for the synthesis to synthesize them and degrade them. Now those enzymes should be localized in the same pathway that contain the neurotransmitter. So it's not something that it's being synthesized elsewhere and then pick up by the neurons and so on. Okay, so there should be uptake system or degradation system for the neurotransmitters. So this one is actually more like to remove the neurotransmitter quickly. As mentioned earlier, if the neurotransmitter is not removed, then the transmission will continue, and then the next transmission cannot occur. Now that should be it should be released in a calcium-dependent manner from the nerve terminal following potassium stimulations. Now this one is more like when we talk about the how the action potential lead to the release of neurotransmitter, then you'll know basically it's the arrival of the action potential at the terminal, then that causes changes in the calcium, and then that causes the release of the neurotransmitter from the synaptic vesicle. Okay, so we will talk about that in a more detail, possibly next week. Physiological, so the substance should mimic the nerve stimulation when applied exogenously. One of the examples that we talk about is actually, if you still remember, the heart. You know, the second receiving heart, actually the same acetylcholine coming from the first heart actually coming to the terminal, and that actually have the same physiological effect to slow down the heart pumping. Okay. Pharmacological. Now, pharmacological is actually more like, you know, drugs or agents that can mimic the action of the proposed neurotransmitters. Okay. Now, one of the example is actually in the case of the acetylcholine, you know, nicotine. So nicotine. Do you know what is nicotine? Anyone? When we talk about nicotine, yes, from cigarette, smoke, yes, good. Addictive, yes, cigarette. So from the cigarette. Now nicotine, that one is the same. Now nicotine can actually bind to some of the acetylcholine receptor. Okay, so there's a pharmacological agents that actually can mimic the effect of the acetylcholine actually in the body. Okay, so when we talk about acetylcholine, we will talk about it in more detail, and that's good, very good. That's also why the nicotine is related to addiction as well, because actually it has a psychological effect. So it should be possible to find specific agents to interfere with the synthesis, release, or the receptor binding. So we will talk about those in more detail when we talk about individual neurotransmitters. Okay, that's actually also a very important ideas that behind how different drugs work. For example, Botox. You know Botox? Anyone? Plastic surgery, very good. Very good. Actually, Botox is makes you look better. I don't know. That's true. Now, Botox actually is a protein that can. Yes, that's true. Like young Lisa. So Botox actually no, it's not killing cell. Actually, it's a protein that can interfere with the transmission of the nerve terminal. Okay, so when we talk about, you know, no, it's not eating the face cell. So it's not eating the cell actually. No, actually, the Botox actually stop the muscle from contractions. And then if you stop the contractions, then the wrinkles will disappear. It's not because wrinkles usually when your face muscle contract when you're in your old age, then you see the wrinkles. Now the Botox actually prevent the muscle from contraction. So we will talk more about it when we talk about the synaptic transmission next week. The details about synaptic transmission. Okay. Very interesting, actually. After this course, I hope that you know more about how those functions and why injection of Botox can be dangerous if it is not performed. It is not performed by doctors. Okay. Okay. Yes. Good. So now you know Botox actually affecting the nervous system. Actually, now so the other classifications of the neurotransmitters including global transmit. It can be generated in 3 general classes: global, specific, and modulatory. Okay, again, now global are the one that are the amino acid type, the GABA and the glutamate. Both are the very important neurotransmitter that actually control your cognitive functions. So it is at the highest concentrations, and it is widely located in the nervous system. The function includes the excitatory or inhibitory. Basically, glutamate is excitatory, GABA is inhibitory. Okay. If it is too stimulate excitatory too much, that will cause seizure. Okay. Seizure is the one that are very strong effect. Okay. Coma is you lost consciousness. Okay, so that is actually depressions. Actually, some of the drugs that act on the GABA can make people unconscious at once. When we talk about the GABA, then we'll talk about those actually. So-called the day, some of the drugs called day rate drugs. Those are actually acting on the GABA. Biogenic amines and peptides, and also acetylcholine as well. Now, acetylcholine actually are more special, in which in the peripheral that cause the contraction of the skeletal muscle, and also in the parasympathetic system that control the different organs as well, for example, the heart. But in the central nervous system, that actually there's evidence that suggests that they control the no, actually, one of your classmates asked if GABA is inhibitory. How does it lead to the unconsciousness when a GABA action is inhibited? No, but actually, when GABA function is enhanced, and the cause unconsciousness. Okay. Now, if the GABA action is inhibited, that might cause seizure. Actually, it's the opposite. Okay. When we talk about GABA, then you know more actually. Biogenic amines, so they are also high, but they are in restricted location. They are more specific actually. As I mentioned, acetylcholine has been implicated in memory as well. And dopamine, one of your classmates mentioned about that actually also involved in addictions. So when we do the group projects, then you'll know more. Modulatory peptide. So one of your students asked, what is the effect of the acetylcholine in central nervous system? Actually, in the central nervous system, the effect is less clear. Yeah. Okay, good. So you know somebody suffer from seizure. No, they are actually not unconscious. Well, they lost consciousness because the neurons are too activated, and so the unconscious that the unconsciousness I mean is actually loss of actions. Okay, but seizure actually is the brain. The neurons are increasing the activity so much that actually the brain is not functioning anymore, and then if you have those typical rapid movement, and then he will not know what happens. Okay, so very good actually. So but it's the one that I mean unconsciousness, like the person just like sleeping, not moving at all. Those are the unconsciousness that I mean, you know, but you're right. You know, when they have suffer from seizure, they don't know what they are doing, but they're still moving actually, but it's moving too much. Okay. Modulatory peptide, they are in low concentrations. They're restricted also. When we talk about the neuropeptides, then we can discuss more about it. Is it okay? So you see, a lot of you now know more about, you know, how the nervous system involved in the things and people around you. So that's what I want you to get. So why the neurons are overactivated would paralyze the system. How do I say it? So just imagine that, you know, the movement and let's say, let's give you an example. So for example, if you just like muscle cramp, you know, so muscle cramp, when it's not necessary because of that, but just think about it. If the muscle is activating too much, then the muscle will not be functioning anymore because it's like in a cramp situation. Okay. The nervous system is the same. If you want to, if the system needs to be functioned properly, they need to be at sequential outcome. But if you just like crazily just activating everywhere, so every neurons are firing, then you lost control. Okay, so that's the reason why it becomes, you know, activating too much, then that becomes seizure. Maybe the next time I find some video or find some well, let's see whether I can find some video that show that what does it mean. Okay, but they're good actually. You're asking all the very interesting related questions. So the global neurotransmitters, so they are functioning in the overall excitatory and inhibitory tone of the CNS. Okay, so if that is excited too much, that will associate with general excitations or seizures. Okay. Certainly, if you are very excited, that's increase your consciousness as well. But if too much, then that will become seizures. Now, inhibitory will result into depressions or coma. Okay, so maybe coma is a better term than unconsciousness. Okay, so coma is a better term. Okay, so in the seizure example is not in coma. Okay, so those are the GABA and glutamate are the global neurotransmitters. Now, specific neurotransmitters, the biogenic amine and peptides, they are found in the more restricted location and associated with specific group of neurons called nuclei. They're generally not found in high concentrations. When we talk about those pathways, then you know that they are located in specialized regions, and we'll talk more about those functions as well. Now, I will say that, you know, those actually are setting the tone of the nervous system. So the global transmitter is the working one. I always give this example is that, you know, you can work, or you can, for example, you can eat, but you can be happy eating happily, or eating sadly. Okay, so what makes the difference? You can still perform the eating process, but your mood may be different. Okay, that's the reason why that one of your classmates talk about the dopamine and serotonin can make you happy or addictive as well. So we'll talk more about those when we talk about dopamine and serotonin as well. Okay. Modulator neurotransmitter. So we mentioned about the axon to axon exosonic synapse. This is one of the example in which that this axon terminal can affect biogenic amines. We'll talk about biogenic amines type of neurotransmitter when the time that we talk about. It's a group of amines. Okay, biogenic amines. So the amines, a group of amines that have effects on the neurotransmitter, in the nervous system. So here is the modulator axon. So usually some of those are peptides. They modulate the effect of the other neurotransmitter. This is the good example. So this neurotransmitter can affect the release of neurotransmitter in this terminal. Okay. Usually they have long-lasting effect because sometimes they actually even can modulate the gene transcription. So those are the modulatory neurotransmitters. When we talk about those, particular, for example, dopamine or serotonin, then we'll talk more in details. It's okay? So we will stop here today. Second. Oh. Yes, so this one is the second level neurotransmitters. This is the first level and affecting this release of this one. Axonal. Let me see. No, this is okay. This one actually modulates the synapse of this one. Synapse of this one. Yeah, this one is axonal. This is one of the examples. So this terminal can affect the to modulate the terminal of this one. Okay. Okay. Any other questions? Any more? If no, then we end the class today so that you can prepare for the next class if needed. So next week, we will spend some time to talk about the project actually. Okay. So if not, then thank you and bye. See you. How is neuromodulator and modulator? Oh, so that's different actually. Neuromodulator can be, for example, a hormone. For example, as we mentioned, the steroid and some of the cytokine can act as a neuromodulator. just a moment. The modulatory neurotransmitters actually is neurotransmitter can function as a can function as a modulator. Language actually involves more than that. Actually, not just for no. Okay. The PowerPoint missed a table in page 16. This one? This table here. Well, maybe I will upload this file to the campus again, so you can download this version. I don't know what happens to the one that in the campus, I guess. Okay. Okay, if no further questions, then we'll stop here. Okay. See you guys next week. Okay. Bye bye.